Okay, hey guys. I'm going to go over the study guide that I sent out to you earlier today. This video will be going over the shapes of bacteria, the Barr-Epstein virus, peptic ulcers, and the lab requisition form and the lab results form. If you want to cover terminology, I would recommend going to your study guide assignments and just reviewing the vocabulary sections at the beginning of those assignments. I will be making another video about lab tests. So what lab tests we learned about, what they tell us, how to do them, a quick overview, and why a doctor might order them. Not that one. I wonder what that. Well, okay, so actually this assignment, if you remember this one, if you have that assignment, that is a good thing to study. That will be, that'll cover everything I just said. I am also putting together a YouTube playlist on my channel that's going to cover stuff that's on this final. So if you go to my YouTube channel at the home page, if you go to the third section, the created playlists, it's the first playlist. And it will sort of look like this, but there will be more videos. Okay, let's start. All right. Okay. So, bacteria shapes. There are three main shapes. Hopefully this screen doesn't move too much when I'm drawing on it, but we'll see. Let's see. I want a different color. Well, this is fine for now. Okay. We are mainly going to worry about this stuff. I'll briefly cover this, but this is the important stuff for you to know for the final. So first we have our bar-shaped bacteria. They're called bacillus. If you have a single cell of a bar-shaped bacteria, it's called a bacilli. Correction, bacilli. Some people say they look like rods, Tylenol pills or something. We then have our sphere-shaped bacteria called coccus, and if you have a single bacteria that is sphere-shaped, it's called a cocci. These fungi here, they can sometimes look spiral-shaped, sometimes they'll just look like wiggly lines. They're called spirillum, and if you have a single bacteria that is spiral-shaped, it's called a spirilla, not a spirilli. It's spirilla, but you can also just memorize the plural form. Okay. And briefly, if you have two bacteria that are stuck together, you're going to put diplo in front of this word describing the bacteria shape. So if you have two cells that are stuck together that are spheres, that would be called a diplococcus. Diplo is two, coccus is sphere. If you have a chain of bacteria cells stuck together, that is called a strepto formation. So you put that before the word describing the bacteria shape. So if we had a string of sphere-shaped bacteria, that would be called a streptococcus. If you have a cluster of cells, sometimes they have a little bit more of a pattern to this, sometimes they're just kind of randomly clumped. That is going to be called staphylo. So you put staphylo at the beginning of the name describing the shape of the bacteria. So if you have a cluster of spheres, you have staphylococcus. Staphylo for cluster, coccus for sphere. All right, so the lab requisition form. If you remember back to lab when you had to draw blood samples and then put it in a biohazard bag and then fill out a form, fold it twice, and then put it in the outside pocket of that biohazard bag, this is that form. This form tells the lab which tests you want done on your sample. You have to fill out this form for any kind of sample you're going to send to the lab, so that can be blood, urine, feces, swabs from your nose, your throat, somewhere else. If you're ordering a pap smear, you have to fill out a lab requisition form. Let's see. Oh, if you want to read more about this, you can find, I have the third version of the textbook, and you guys probably have the fourth, so I'm not going to give you the page numbers, but if you look around figure 1-6 and 1-7, you'll find information on this form. Important information to have on this form is definitely the patient's name and the patient's date of birth. Those are the two identifiers that are going to be really important in matching the results to the patient later. Some clinics will want you to have the insurance and billing info, some won't. Some forms will have the space for it, some won't. Some forms will also have a space to note any medication that the patient is taking that might interfere with lab test results. Always indicate whether or not the patient is fasting. Even if you're doing a test that it doesn't really matter, check it off. You will tell them what tests are being ordered by just checking off whatever tests that you want, just like you did in lab. You'll need the physician's information on your form and the date and the time that the specimen was collected. That way, if the specimens are delayed in getting to the lab, maybe the courier that takes the specimens from your clinic to your offsite lab got delayed for some reason. If there's been too much time between the collection and when the procedure for the test is performed, the specimen might not be good anymore, and the results will either give us bad information or will not give us any information at all. So always write the date and time that your specimen was collected. And this is sort of what that form will look like. I'm not going to really look closely at it because we didn't use it in lab, but that's kind of like the template that it generally has. Okay, so wow, I can't talk apparently. The lab results form is the paperwork that the lab will send to the clinic when the tests are complete, and it describes the results from the test. And it will tell you whether or not the results are normal. This, if you remember this packet, where you interpreted the results and looked at the scenarios in your competency document, and then had to answer some questions about maybe what this person will be diagnosed with. That's the form that we're talking about. Let's see. You'll be able to find this PDF on Canvas, and if you want to learn more about this form, go to the area near Figure 1.8 in your textbook, and you'll be able to find more information. Always have your two patient identifiers on, they will always have the two patient identifiers on the form, the patient's name and the patient's date of birth. Always match that to the patient's record when you're recording the lab results. The form will list the tests that were completed. So this form right here says that there was a basic metabolic panel done, and a pro-time test done, and a PTT test done. So it just describes what tests were completed. It will also tell you what normal ranges you would normally expect for that test, what units the test is reported in, and the actual test result that the patient got. So if you look over here, if the patient's results is in range, it will be in that column. If the patient's results is out of range, it will be in that column. And you know what the range is because they list it over here in this column. And these will be the the units that you record the results in the patient's medical file. The lab form will say the date and time that the lab got the specimens, and then the date and time when the specimens were tested. Sometimes they're different. And they'll also have the physician's name and information on this form, so the lab knows where to send this, where to send the results after they're completed. All right, so if you remember back to the beginning of class, the beginning of the quarter, we examined urine casts and crystals underneath a microscope. You're going to need to remember how to calculate the total magnification that you are using to examine a specimen. So when you're looking at a specimen right here, the light travels up from the light source through the objective lenses, whatever lens you have in place. It'll go through the objective lens and then out through the eyepiece. And as it goes through the eyepiece, it'll cross through an ocular lens. Both the ocular lens and the objective lens you're using have different magnifications. The ocular lens usually has a magnification of 10. When you see X after a number, that just means magnification. So you would read this as magnification of 10. So these are the eyepieces. That's what they look like close up, and they magnify the image by 10. A microscope will have 3 to 4 objective lenses. I'm trying to remember which one they usually have. I feel like if they don't have the 4th one, they usually don't have this one. They'll have these guys. But if you have 4 objective lenses on your microscope, you'll have these 4 right here. You know the magnification by looking at these numbers right here on your lenses. The lower power lens, the red, will generally have 4 magnification. Your medium power lens, the yellow, will have a magnification of 10. High power objective lenses have a magnification of 40. And then your oil immersion lenses, so that's when you put that little drop of oil on top of your cover slip, have a magnification of 100 times. So when you are calculating the total magnification, you want to multiply the magnification of the ocular lens by the magnification of the objective lens that you're using. So let's say that I'm observing a specimen using the low power objective. The low power objective has a magnification of 4, and that it's always going to go through the eyepiece. So you're going to multiply it by a magnification of 10, and then so your total magnification is 40x. I just edited out a blooper. I had a massive brain fart, so onwards. You take the magnification of the ocular lens, your 10x right there, and you multiply it by the magnification of the oil immersion lens, which is 100x, and your result is 1000x. So when you're looking through the oil immersion objective lens, you would say that the image is magnified 1000 times. And if you had to write this down somewhere, you would just write it as 1000x as shorthand. Okay, I think that's enough doodling on this page. Okay, let's get through the rest of this really quickly because I'm sure you're getting bored. What the heck is mono? Mono is short for mononucleosis, and mononucleosis is the name of the disease that we're observing here. Symptoms generally are fever, rash, fatigue, swollen lymph nodes, swollen glands. Mono has a very large range of symptoms. You don't need to memorize the symptoms. Just know that mono is short for mononucleosis, and that is the disease. That is what's going on in your body. Mononucleosis is caused by the Barr-Epstein virus. The Barr-Epstein virus is the organism in your body that is causing the disease of mononucleosis. H. pylori is a bacteria. The Epstein-Barr virus is a virus. This is a bacteria. H. pylori is short for Heliobacter pylori, but you don't have to memorize that. Just know that H. pylori is the disease-causing agent that we're talking about right here. And that the condition in the body that H. pylori causes is peptic ulcers. It can cause a lot of different diseases, but the most common one is peptic ulcers. Peptic ulcers can happen in the stomach or can happen in that little section right after the stomach called the duodenum. So that's why you say peptic and not necessarily just stomach ulcers. What happens is as H. pylori is growing inside the stomach, it will start to eat away at the mucus lining inside of your intestines, and that mucus lining is protecting that smooth muscle that lines your intestines, and then once that mucus is gone, the bacteria creates a lesion, an open wound, and then that causes blood to leak into the stomach and stomach acid to get into that wound, and it's really nasty. We say that it causes a rupture in the stomach and duodenal lining. There are other causes of stomach ulcers or duodenal ulcers, but H. pylori is the most common cause. So remember that H. pylori most commonly causes peptic ulcers, and peptic ulcers are most commonly caused by H. pylori. Okay, I think that's it. I'm going to go work on that video about lab results and stuff and uses, and I hope this has been helpful. Okay, bye guys.